Hello students, welcome back. In this session, we'll be discussing about the important method of gene transfer by using the chemicals called as chemical methods of gene transfer. As the name itself indicates, in this method we use some chemicals in order to aid or in order to accelerate the transfer of gene into the recipient cells. Some of the chemicals are used here, such as PAG, that is polyethylene glycol, or DEAE, or the chemicals such as calcium chloride or calcium phosphate. Mainly these three chemicals are used to transfer the gene into the cells. First, let's discuss about the first method in chemical methods of gene transfer, that is PEG mediated transformation or the PEG mediated transformation. Before we start this, what is the importance of these chemicals is, whenever we are transferring the DNA or any gene into any other recipient cell, the first boundary for the transfer of the gene is the cell membrane. That is, whenever you are transferring a gene into a cell, the cell is lined by the cell membrane. This cell membrane is mainly made up of lipids, which you know, and this cell membrane is hydrophobic in nature and is negatively charged. Whereas against this negatively charged hydrophobic cell membrane, you are transferring the gene or the DNA which is hydrophilic in nature and is negatively charged. The difference is that the negatively charged hydrophilic DNA has to pass across the negatively charged hydrophobic cell membrane, which is a tough task for delivering the gene into the cell. In order to transfer the DNA, in this method what is done, the cell membrane is made to interact with some chemicals which we are going to call it as synthetic complex. Synthetic complex means DNA is mixed with some chemicals which we are going to call it as the complex of DNA and the chemicals, or you are going to call it as the synthetic complex, so that the DNA which is having negatively charged must be converted into a positively charged molecule, so that it can easily interact with the negatively charged cell membrane and can pass across the cell membrane. This is the principle behind using the chemicals in order to accelerate the transferring the negatively charged hydrophilic DNA across the negatively charged hydrophobic cell membrane into the cell. So first let's discuss about the first method of gene transfer using the chemical. The chemical used here is the polyethylene glycol. Polyethylene glycol is nothing but it is a chemical which is used to transfer the DNA. Especially in this case, what is done, in a test tube, the DNA which you want to transfer must be taken, and to that test tube we need to add the divalent cations such as calcium along with the polyethylene glycol. The percentage of the polyethylene glycol usually used is 40%, which is dissolved in mannitol, and this mixture must be incubated so that the protoplast can be easily transformed. Protoplast means, you know, the cell which is lacking the cell wall, which is having only the cell membrane. So whenever we are adding the polyethylene glycol and the calcium along with the gene or the DNA, what happens, the polyethylene glycol is going to form a complex with the DNA, and whatever the calcium ions which we have added in the test tube is going to form the pores across the membrane, and through the pores whatever the gene or the DNA which we have added can be easily transferred into the protoplast, and thus the cell is said to be transformed. So this is the first method of chemical gene transfer using the PEG. The second method of gene transfer is DEAE, that is dextran mediated transfection. The word transfection can be split into transformation and infection. It's very simple. By infecting a cell with a DNA, so transfection means you are infecting a cell. Infecting a cell means you are transferring a foreign gene or foreign DNA into a cell, so that the resultant cell would be called as and transformed with the desired gene or the gene which you want to transfer into that cell. It could be done with the help of another chemical that is called as DEAE dextran. It's also called as diethyl aminoethyl dextran. The importance of this DEAE is that it is the first soluble polycationic carbohydrate which is used in gene delivery studied. This DEAE forms aggregates with the DNA through electrostatic interaction. So I am going to repeat, whatever this DEAE is there, it is a soluble carbohydrate which has got the positive charge. I say it as what it is, a cationic carbohydrate. This DEAE is going to combine with the DNA. The DNA is negatively charged through the interaction that is called as the electrostatic interaction. The entire complex, the complex is formed by the aggregation of the DEA with the DNA, and this complex will gain a net positive charge. Negative charge DNA with the cationic DAE is going to gain a net positive charge. This complex which has got the positive charge, complex means DEAE and your desired gene which you want to transfer, now has got the positive charge. So this positively charged complex, now we need to allow it to interact with the negatively charged cell membrane, so that the interaction occurs between the positively charged complex and negatively charged cell membrane to promote the uptake of the DNA through the process that is called as the endocytosis. That is, in liposome mediated transfection, you have studied how the liposomes are eaten or taken up by the cell membrane. In the same way, through the endocytosis process, the positively charged complex which is formed by DEAE and the DNA or the gene which you want to transfer is taken into the cell or the protoplast through the process that is called as the endocytosis, and thus the cell is said to be transformed. And this is how the transformation is achieved with the help of the DEAE, and this method is called as dextran mediated transfection. So this is the second method of gene transfer. The third method or the third chemical used for the transformation purpose is the calcium phosphate mediated DNA transfer or the calcium chloride mediated DNA transfer. In this aspect, what is done, the main chemical used is the calcium phosphate, which is mainly made up of the divalent cation, that is the calcium. Especially this method is followed whenever we are transferring the gene in case of the bacteria, or especially in bacterial transformation, we need to use the chemical that is called as the calcium chloride or the calcium phosphate. So in this case, what is done, the cell into which you want to transfer the DNA must be taken in a tube. To that tube we need to add the calcium chloride solution or the calcium phosphate solution. Then to this tube we need to take the gene or the DNA which you want to transfer. That means totally the test tube, in the test tube you need to take the host or the bacterial cell, plus you are adding the calcium phosphate or calcium chloride solution, plus you are adding your gene of interest which you want to transfer. So totally 3 components you need to take this, and you need to incubate it at about temperature 42 degree Celsius for some time, and later immediately the tube must be lifted from the 42 degrees Celsius bath, and immediately it must be transferred to the ice whose temperature is about 0 to 4 degrees Celsius. So in this way, or this treatment is called as heat shock treatment, where suddenly you have altered the temperature from high temperature to lower temperature. That is what you have done here. You have transferred the test tube from the high temperature, say about 42 degrees Celsius, to low temperature of about 0 to 4 degree Celsius. So this kind of treatment is called as the heat shock, and this heat shock results in destabilization of the cell membrane. And as we have already added divalent calcium ion to that tube, it is going to cause the development of the pores across the membrane, making the cell membrane porous. So through this porous cell membrane, the gene or the DNA which we have added will migrate into the host cells, which will result in the transformation. This is how, by using the calcium chloride, the DNA transfer could be done in case of the bacteria. So these three are the important methods of gene transfer in case of, or these are the methods of gene transfer. Thank you.